today we're going to finish up our PowerPoint on water balance and hopefully get into glomerular filtration. So just a recap, last time when we talked about the establishment of the medullary osmotic gradient. We looked at the role of our sodium potassium pump in the proximal tubule. We looked at the structure of the loop of Henle, as well as the structure of the vasa recta here, all of which is creating this medullary osmotic gradient where we have lower osmolarity at kind of our baseline level of 300 in the cortex and in the medulla. And then as we go deeper into the medulla, we see increasing osmolarity up to about 1400 milliosmoles. So when we compare the osmolarity of filtrate, this is blood flow, but it's also true for filtrate, we have relatively lower osmolarity in the tubule versus in the surrounding parts of the medulla. So that's our setup for today, when we're going to start thinking about the influence of hormones that allow us to control the amount of water absorption in the kidney. Okay. So when we talked before, we talked about the water being reabsorbed in the proximal tubule. And we saw that that water absorbed in the proximal tubule, or reabsorbed in the proximal tubule, I should say, is just following the movement of ions, chiefly sodium. So we are using a sodium potassium pump ultimately to draw sodium out of the filtrate and ultimately into the plasma. And because those ions are moving, as they move, they increase the osmolarity of the regions they are in as they are traveling. So they increase, particularly in the peritubular interstitial fluid. So they're like fluids surrounding our tubular epithelial cells, but on the opposite side from the filtrate. Anyway, so we increase osmolarity farther away from the filtrate, draws water towards it because osmolarity, and that's a pull on water. So that was the proximal tubule. We mentioned that this isn't a regulated process, like we didn't see any hormones coming in and turning on and off receptors or anything. This is just happening all the time. It's just happening because of this movement of the ions. It's not really a toggle switch, it's just bulk reabsorption. Now that we're moving on to what's going on in the distal tubule and the collecting ducts, we're going to see that this reabsorption is regulated. So we can control how much water gets reabsorbed in the distal tubule and in the collecting duct. And this is good for us because this means that we can change how much water we're keeping in our system, in our body, versus how much water we're treating as urine, in accordance with how much fluid we've taken in and how much fluid potentially we produce through metabolism. So we want to be able to control this because we're really using the kidney processing blood and fluid in order to maintain appropriate blood volume so we can maintain blood pressure. All right, so that's happening in the distal tubule and the collecting duct. We're going to be talking about the role of this hormone ADH. That's like a preview for what's coming up here. We did this brief review of the osmotic gradient in the medulla, which is established by the countercurrent multiplier, which is just a specific term for the fact that that flow on either side of our loop of Henle is in opposite directions. To understand why ADH is able to regulate the reabsorption of water, we needed that all kind of a setup so that we understand what's going on in the medulla as ADH is sort of turning on and turning off our ability to reabsorb water. Okay. So as we look here in the distal tubule and in the collecting duct, we're going to see what we're doing in order to regulate, in order to control the reabsorption of water, is we're changing the permeability of our epithelium. So when we talked about the loop of Henle, we talked about the fact that our descending part was permeable to water, but our ascending part was impermeable to water. So hopefully we've already keyed in on the fact that when we're thinking about the nephron, we do want to think about some places water can cross, some places water can't cross. Okay. So I'm going to briefly draw just a cell so that we can understand that these two comments about the presence of these aquaporins. Okay. So whenever we're talking about a cell, we want to think about where the blood is. And in the case of a nephron or anything with a lumen, we want to think about where that lumen is. So here's going to be a blood vessel, potentially. So the reason we're keying in on that is because that tells us where our basal and therefore our basolateral membrane is. So like the bottom of the cell. All right, so that's our basolateral membrane for the base, and then the sides are the lateral part, right? And then we have our apical membrane, often draw kind of wiggly. And that's where we're going to be neighboring the filtrate. So I'll draw this sort of filtrate arrow in orange. Then there's more cells all around, right? Okay. So what we're doing with ADH is controlling the permeability of this cell, this epithelial cell that we're focusing in on. And the way we do that is we control where and how many aquaporins we have present. So there is always an aquaporin 3 on the basolateral membrane. And I just drew it all the way across so you can think of the water going all the way across. It's not physically attached to the bloodstream. So aquaporin 3 is always present on the basolateral membrane, which means that if our gradients are right, we can always have the movement of water from the inside of our epithelial cell into the plasma. So that's always possible, assuming we have the right concentration gradient. Okay. But what we're going to be changing is the presence of aquaporin 2 here on the apical side. So this is aquaporin 2. So if we're thinking about the movement of water out of the filtrate, so we're thinking about reabsorption back into the bloodstream, it doesn't really matter to us. It doesn't just matter that we can get water from the inside of the cell into the plasma, because we need water to get all the way from the filtrate into the plasma, right? We need it to go first out of our filtrate, so the stuff that's becoming urine, into the cell, and then also all the way into the bloodstream. So if we do not have this aquaporin 2 present on the apical membrane, water can't get into this epithelial cell, and therefore it doesn't cross into the plasma, right? Then our epithelial cell and the plasma essentially just come to equilibrium with each other, right? And we don't have any of this water moving, right? So the way we control whether water moves all the way across is by controlling how much aquaporin 2 there is. So aquaporin 2 is only going to get inserted into that apical membrane, so it's only going to be present when we have ADH swimming around in our bloodstream. So ADH is our hormone that's controlling the presence of aquaporin 2 on the apical membrane. So when we have ADH, we will be able to reabsorb water because we'll have both of these aquaporins present. But when we don't have ADH, we won't have any aquaporin 2, so we won't see movement of water in the distal tubule or the collecting duct. We would still see the bulk reabsorption of water in the proximal tubule. That's not affected by the ADH. So when the membrane of the late distal tubule or the collecting duct is impermeable to water, so meaning we don't have that aquaporin 2 there, That means that water can't leave the tubules because it can't get into the cells surrounding the tubules, which means that no water is moving from that filtrate into the plasma. Which means that that water is staying in the filtrate and that water is therefore staying in the urine, right? So if we're not moving it into the blood, it stays just right where it is, which means that more water is excreted in the urine when we don't have the ability to move water back into the blood, when we don't have these aquaporins. So when we don't have ADH, when ADH is not present, that means that you'll have more dilute urine and probably more urine because that fluid is just staying in the tubules and ultimately in the urine. And when we do have ADH, we do have this hormone present, we insert the aquaporin tubes into the apical membrane. And this means that water can flow all the way across the cell. So it flows into the cell, and then because that aquaporin 3 on the basal membrane is always there, into the plasma. And because we're moving this water, essentially what we're doing is concentrating the urine, right? So it's going to get like darker, probably you'll have less volume of it because you're reabsorbing the water. You're pulling that water into the blood. You're probably trying to maintain blood volume, something like that, right? Trying to not get too dehydrated. So this is concentrating your urine there when we have ADH present. The maximum amount of water that gets reabsorbed depends on the length of the loop of Henle. So basically like how deep into the medulla we can go and how concentrated that medulla gets. And it's mainly a sort of comparative comment because in some animals like camels have creepy long loops of Henle, and that's part of how they can conserve water so well, is that they can really soak up all that water back into their blood, so it doesn't matter that they don't drink so much. Okay. For us, the maximum urine concentration, so the maximum osmolarity that we're going to see in the urine is 1400 milliosmoles. So you can't just like, if you think about it, this is obvious, right? You don't ever just pee out like bricks of salt, right? Like that's not a thing that happens. There is a certain amount of liquid that you need to keep in urine in order to actually pass out the wastes. So at that minimum amount of liquid remaining in the urine is at 1400 milliosmoles. So the reason this can happen if we're looking at kind of the structure of the nephron and the surrounding cortex and medulla. So we want to remember, all right, our cortex is at 300 milliosmoles. Our medulla is going all the way down to 1400. Okay. And our tubular fluid coming out of the loop of Henle, right? So our loop went down, we're back up. So now we're in the distal tubule. Remember, we have a particularly low osmolarity of the fluid in the late distal tubule, right? That was one of our numbers that we were remembering, right? 300 was our normal amount, 1400 was our maximum amount deep in the medulla, and the other number I told you you'd probably want to remember was this 100. So what we're going to notice is this means that there is a pull on water from the cortex, right? 100 is lower than 300, meaning that we have a pull, we have a force to move water into the cortex from the distal tubule. But if we are impermeable to water, doesn't matter how much force there is. If we don't have a way to get across the membrane, we can't get across the membrane, right? So as we continue to pass into the medulla, we can see that the force is getting stronger and stronger, right? These numbers are getting farther and farther apart. But again, we're looking here on the left at a situation where these regions are impermeable to water. So even though there is a pull on the water, nothing's happening because we don't have a way across the cells. Okay? So our tubular fluid is just staying at 100 because no water here is getting reabsorbed. Okay. So the urine is going to have very low osmolarity. And it'll probably be high in volume because all that water stayed in there. So there's just more stuff, right? It's really the water that's creating the volume of the urine. On the right-hand side, what we're looking at is what happens if we do have permeability, right? In this case, we're thinking like we do have ADH running around in your bloodstream. So we're inserting those aquaporin tubes, which means that we've made the distal tubule and the collecting duct permeable to water. So now, because we have both of those aquaporins, right, we have them on both sides of our membrane, we are able to move water all the way across from that tubular fluid that has a lower osmolarity than the higher osmolarity here in the cortex. Right, so remember, osmolarity as it goes up, basically that's indicating sort of higher concentration of solutes, right, which exert a pull on water to come water them down. Okay, so we see movement from the tubular fluid to the cortex from the distal tubule. So we can see that that increased the osmolarity of the tubular fluid a bit. And as we continue through the collecting duct is going through the medulla as it goes to the minor and major calyces, the renal pelvis, ultimately the ureter, the bladder, and out the urethra. So we can see here that it's increasing, but we also have increasing osmolarity of the surrounding medulla. So we see we continue to have the movement of water out of the fluid in the collecting duct, which is how we're ending up with that maximum osmolarity, right? So this is the deepest point of our medulla. So we can come to equilibrium essentially with that deepest point of the medulla. So this would be our high osmolarity urine, which would be low in volume because we took all that water out of the fluid and we put it back into the bloodstream. Okay, so we are going to take a zoomed in look at the process of how these aquaporins are inserted, but questions on this basic concept first. Okay, so this is really why we were focusing on what's up with this medullary osmotic gradient, like remembering that it exists and why it exists, is because we're setting up this situation where we can reabsorb water if we have ADH, which is our antidiuretic hormone that allows us to reabsorb water. And that was kind of the whole point of that setup. So we do have an obligatory water loss. There's a minimum amount of water that you have to excrete in the urine per day. And this is because your cells are producing all sorts of waste products. You also do want to be constantly balancing the ions, making sure that the concentration in your blood stays the same. Right? So that maximum osmolarity in the urine reflects the osmolarity that we see in the medulla. So for us, it's 1400 milliosmoles. Make sure that we can excrete some solute so that we don't have it just building up and building up and building up. The minimum water loss that corresponds to that maximum osmolarity and that minimum water that has to be excreted is about 440 milliliters per day. So that's the necessary amount that's going to allow you to eliminate all the solutes you're not reabsorbing, but that you are producing by metabolism, for example. So things that we have to excrete in order to keep our body functioning appropriately. These are just some questions for later. So now we're going to take a zoomed in look at that antidiuretic hormone. Okay. The antidiuretic hormone, ADH, is controlling the water reabsorption because it's controlling the permeability of those two regions we were just looking at, the late distal tubule and the collecting duct. It is kind of like a dimmer switch, right? So urine osmolarity can be anywhere between 100 milliosmoles, right? If these tubules are absolutely not permeable, that's what we saw the osmolarity of the fluid coming out of the loop of Henle was. So if we can't move any water at all out of that anymore, we're at 100 millismol. So that was that left-hand image that we just looked at. Or our maximum, we move as much water as possible out, we're going to be at 1400 millismol. And it's all because of those aquaporin 2s that are getting varied by ADH. So ADH we call anti-diuretic hormone. That's what it stands for, right? Anti-diuretic, and then I got hormone. Right, so a diuretic, right, is something that makes you pee a lot, right? So high volume of urine. ADH is anti-diuretic, right? So it's going to lower the volume of urine. And the reason it's doing that is you're reabsorbing the water. That's how you fight against, right? Like a diuretic. Sorry, that wasn't very clear. It's like the opposite of being in this state where you're having high volumes of urine, right? So a diuretic would make you more impermeable. The water has to stay in the tubule, high volume. If we have ADH, we're removing the water, putting it into the blood. So it's the opposite effect, which is why it's anti-diuretic. Okay. So here's what we have happening if we look at the cell specifically. So we can see this is that same cell that I was drawing before. So here's our paratubular capillary, right? So that's the paratubular capillary right next to our tubule. So that's the blood, the plasma we're going to be moving water into. Here's the lumen of our late distal tubule, or our collecting duct, right? So this is where we have our soon to be urine flowing. And this is our cell, specifically a principal cell in this tubule. We'll go through it piece by piece. All right. So the first thing we're keying in on is that we have ADH present. So ADH is our hormone, right? So what that means, right, is that we've produced this hormone, we've produced this molecule somewhere else. Part of our endocrine system, we put it into the bloodstream, right? Because we're trying to send a signal to somewhere else in the body. In this case, we're sending our signal to the kidneys. So just like with other cellular signaling, the first thing that happens is that the ADH migrates out of the blood and it binds onto a receptor. So in this case, an ADH receptor, which happens to be a G protein coupled receptor. We have ADH in the blood. It binds to a receptor on the basolateral membrane of this principal cell that's either in the late distal tubule or the collecting duct. So the next thing that happens, G protein receptors in general work out by breaking apart that G protein. Then we're going to send a signal basically into a sort of second messenger system, right? So in this case, we happen to be activating this other membrane protein, adenylate cyclase, which tells the cell to start making cyclic AMP. Right? So cyclic AMP is just a very common sort of second messenger that we see in our cells. Right? So we're kind of passing the signal. We're just like telling this cell, hey, there's ADH in our blood. Okay? There's a signaling pathway. In this case, it happens to be through protein kinase A. Okay. And what happens after this signaling transduction pathway, so we always have these aquaporin 2s hanging out in vesicles. So not in the membrane itself, but inside the cell. And a vesicle basically is like a little baggie of membrane that's floating around in the cytoplasm. In this case, our little baggie of membrane has aquaporin 2, just waiting for ADH to be present. Okay. So when we get the signal, when we have ADH in the blood, it activates our receptor, it activates our second messenger system. This tells this vesicle to go insert itself onto the apical membrane. So the way membranes work, right, is like basically when they bump into each other, they just kind of become one thing, right? So they break open. And in this case, because that vesicle had these aquaporin 2s in that membrane, now the aquaporin 2s are on the apical membrane. And this means water can now get into this cell. So basic steps here. There are a lot of named molecules here, but I'll focus in on what I'd actually like you to know. So what I want you to know is ADH present. And it's really just what I've been saying out loud. 2, there's a receptor. 3, there's a second messenger system. And we can skip some of these details, but we do want to know 4 vesicles with the aquaporin 2 are inserted onto the apical membrane. Those are kind of our steps here, right? So we noted the presence of ADH. It hit the ADH receptor on our cell. So now we know that we have ADH. We want to reabsorb water. This message gets passed along. The place where we have our aquaporin 2s hanging out at the ready is this vesicle, and they're now inserted on the membrane itself, creating a permeable membrane, a membrane that's fully permeable to water across both ends. Okay. So the regulation, or ADH itself, our antidiuretic hormone, also gets regulated. Right, so your body does control whether it's secreting ADH. It wouldn't be very useful to us if we were just secreting it the same amount all the time. So ADH is coming from a place that's hopefully vaguely familiar to us, at least in its name, right? So in A&P 1, we talked about the hypothalamus. Right, so the hypothalamus, we spent a lot of time talking about the anterior pituitary and less so the posterior pituitary that are kind of hanging down below the hypothalamus. Right, we looked at the fact that posterior pituitary, we just have those neurons extending into the posterior pituitary versus the anterior pituitary. We talked about this portal system. Okay. Anyway, so when I say that we're releasing it from terminals in the posterior pituitary, we're referring to that little hangy dangly bit of the brain, right, coming down below our pituitary glands or coming from the posterior part. Okay. So our ADH is originally coming from the terminals in that posterior pituitary, but we want to control how much or whether we're releasing ADH. So we also have a receptor in the brain that senses the osmolarity of the blood. So it senses basically solute concentration in the blood. So it knows how salty your blood is getting, basically, right? Like whether you need to bring more water in to maintain appropriate concentrations or whether you've already got plenty of water. So those receptors are osmoreceptors because they're sensing osmolarity. And they happen to be in a place called the organum vasculosum of the laminae terminalis. Okay, so we just call it OVAL-T, and every time I see this acronym, I think about OVAL-T. I don't know why. But they're sensing osmolarity, right? So they're sensing your plasma, sensing the solutes, which is going to tell us whether we want more water or not. So what's true about this osmoreceptor, that even though it's in the brain, it's not surrounded by the blood-brain barrier, which is why we're able to sense the osmolarity. It's why we're able to tell how much of our solutes are there in the plasma. Otherwise, we'd be kind of blocked. So we like to protect the nervous system. We like to protect the brain with that blood-brain barrier. Okay. So that's one way, basically, we're regulating, is by sensing osmolarity. Okay. there's a second thing we're doing, which is ADH is also going to be affected by our baroreceptors. So when we were talking about the cardiovascular system, we said that we have baroreceptors that can tell what our blood pressure is and what our blood volume is, basically based on the stretch in the walls of our blood vessels. Okay. So if we have low baroreceptor activity, right? So basically, what would this mean about math? Does anybody remember if we have low baroreceptor activity? Yeah, exactly. So this is going to be corresponding to low blood pressure, or low MAP, or low blood volume. Okay. So basically, essentially, we don't have enough blood pressure, we don't have enough blood volume, and a way we can correct this is by increasing the amount of water in the plasma, right? Because blood is mostly plasma, so this is going to increase the blood volume. To do that, we have to reabsorb the water. The place we reabsorb water from is that filtrate in the kidneys. And we do it when we have more ADH, right? So if we have low pressure, low volume, we want to conserve water, we can do that by increasing ADH secretion, which therefore increases water reabsorption. Here's a little flow chart of what that might look like. So the first thing we're going to notice up at the top is the osmolarity of that extracellular fluid has increased, right? So we have a lot of solutes in this fluid that's surrounding our osmoreceptors. And the reason we have high osmolarity there is because there's highly osmolar blood. Okay? So our receptors, our osmoreceptors, are going to detect and respond. And their response is basically increasing the activity of their neurons, which we think of as neurosecretory cells because they also have an endocrine function, basically. Basically, all we mean is they're neurons that secrete hormones, right? So they're secreting specifically ADH. The location for that is our posterior pituitary. ADH swims around in your bloodstream. That's what it means to be part of the endocrine system. That's what it basically means to be a hormone. So once it gets to the kidneys, we insert those aquaporin 2s onto the apical membrane, increasing water reabsorption. So that water is moving into the plasma. And we want to remember the opposite. If it's in the plasma, it's not where it was before, right? So it's not in the tubular fluid anymore. So to maintain blood volume, we're decreasing water excretion, meaning we're conserving body water by keeping it in our bloodstream, keeping up our blood pressure, watering down this high osmolarity that we've sensed up at the top. So this is a negative feedback loop because as we reabsorb water, we decrease the osmolarity and the signal kind of gets turned off. This is just a note also that our blood pressure and our blood volume are also related to this same process. So all that's changed here is what the driver for this effect on the hypothalamus is, right? Like none of our mechanism for how we're reabsorbing more water has changed. All that's different here is that instead of reacting to osmolarity, we're reacting to signals from these baroreceptors that are telling us we have low pressure and low volume. Otherwise, the entire process is the same, and these would probably be going on at the same time. Yeah, this is the one piece that we want to come back to. So this is a note for myself as well as for you. Our next topic is going to be how the nephron starts and how we get that fluid into the nephron in the first place. So we'll come back and talk about the start of this all after we talk about that. Reminder that you looked over the anatomy on Friday so that you have the context for this discussion. Okay. So now we're going to be working in our urinary system renal function packet. Chapter 18. So this is the part that's going to be talking about the basic renal exchange processes, which since we started at a sort of weird place, we've already kind of introduced these and we've talked a little bit about them. Specifically, we've talked a lot about reabsorption of water. And now we're going to focus starting in on filtration as well as secretion. Okay. And at the end of this, we'll touch back again on micturition, which I know you took a look at on Friday on your own, but we'll recap that as well. We're going to start out with basic functions of the urinary system. All right, so we've been talking thus far, really the main job of the kidneys is to regulate. Largely what it's regulating is water. It's actually not a bad thing. We started with water balance. That's is honestly one of the biggest and most important jobs, especially because that affects blood pressure. But we're also regulating other things about the plasma, right? So we're regulating ionic composition, which is kind of the same thing, right, as regulating its osmolarity. Ions are solutes, so increases in ions does increase osmolarity, although we can get pickier depending on whether maybe we're trying to control sodium specifically or potassium specifically, as we will see when we come back to our balance 2 packet. We regulate plasma volume and plasma osmolarity just overall by regulating water, which we've now talked about in detail. We also regulate plasma pH, and we remove metabolic waste products and other things for the plasma. So the urinary system is really filtering the plasma, so filtering the blood, making sure that its composition is what we want and that we have the amount we want as well. There are some sort of tag-on functions of the kidneys as well. So when we were talking about red blood cells, we said that they're being created in response to this hormone erythropoietin. That's coming from the kidneys. So that is another job of the kidneys. They also secrete something called renin, and we will be talking about that later in detail. Activates vitamin D3. Way back in A&P 1, we talked about gluconeogenesis. Right? We talked about how your body can make new glucose when you don't have glucose actively coming into your body, right? So like in our post-absorptive state, we can make new glucose so that we can feed it to nerves, basically. So that's also happening in the kidney. So the structure of the nephron over here, slightly different than the image we gave you for Friday, but we see some of the things you probably drew. Right, so I think we had you look at the glomerulus. Right, I know we've talked about it at least. And this is tracing blood flow to the glomerulus. So afferent arterial, actually let's start at the very beginning. So we have our interlobar artery coming into the kidney, giving off little loops, our arcuate arteries. Coming off of them, we're swirling into a ball. So this one going in is our afferent arterial. A means we're carrying 2 somewhere, just like when we had afferent nerves. Now we have an afferent arterial. Then we have the glomerulus where we're going to see filtrate moving. And then we have our efferent arterial, so moving away. And we can see that that swirls into peritubular capillaries as well as the vasa recta. So there are ways that blood is flowing around other parts of our nephron. So our basic exchange processes are going to be happening between two locations. So we're going to be paying attention to what's going on in the nephron versus what's going on in the neighboring blood flow, which is usually going to be various paratubular capillaries, right? So when we talk about fluid that's going to flow into the nephron, the first step we have, right, is filtration. So filtration is movement from the glomerulus into Bowman's capsule, this sort of head, this kind of mouth piece of the nephron. So we have filtration happening here. That's our filtration. Reabsorption, we've just been talking about a lot with water. Right, so reabsorption is movement from the tubular fluid into these paratubular capillaries, right? So back into the blood. It moves out of the plasma through filtration. Now it's moving back into the plasma from the filtrate. That's why it's reabsorption here. And then secretion. Sometimes we'll see things moving from the paratubular capillaries into the filtrate. So we secrete stuff, squeeze it out into the tubules. And then excretion is out of the body, aka urination for our purposes here. So those are all just terms that are talking about the direction of flow, where these materials are being exchanged. So filtration from the glomerulus into the tubule, and that's the beginning of this whole process in the nephron. Reabsorption from the tubular fluid back into the plasma, secretion from the plasma into the tubular fluid, or excretion out of the body. You see all these happening. We're going to start out with glomerular filtration. So glomerular filtration is the movement of protein-free plasma from the glomerulus into Bowman's capsule, aka everything except for proteins that's in your blood. I mean, not blood cells, but like all the contents of the plasma, other than the proteins, is moving into the nephron. It's moving into the first part of the nephron, into the structure called Bowman's capsule. The rate of this is about 125 milliliters per minute, or 180 liters per day. So this should immediately raise a sort of flag in your mind, right? This is why water reabsorption is such an important thing for the kidneys to do, right? Like if you were peeing out 180 liters a day and removing 180 liters of fluid from your blood every day, I don't know, you'd be dead in like seconds, right? Like that's not sustainable. You don't have that much blood. You don't have anywhere near that much blood, which is why you have to move a lot of that water specifically back into your bloodstream. We're filtering out tons and tons of fluid, just leaving proteins in the plasma at first, and then anything else that we want to save, we're going to reabsorb into the plasma. So there are barriers to the movement of things from the blood in the glomerulus into Bowman's capsule. So we're going to look at the microscopic structure here. So for filtration to happen, for things to move out of the plasma in the glomerulus and into the capsule, they're going to have to pass through fenestrated capillaries. So this is a specialized type of capillary that does let a lot of stuff through, right? When we talked about capillaries before, many of them that we saw, they allowed like gases to move across, but you weren't leaking tons of fluid. Here in the kidneys, we want to be leaking lots of fluid because we're trying to regulate, we're trying to process, we're trying to move waste. Okay. So in these fenestrated capillaries, we're going to see that the barrier for filtration is made up of the layers we have just still existing in the areas where we have these slit pores. So here are our cells, all right? Our cells. We can see that there are some layers that, even though these cells are kind of widely spaced, we do still have a little bit of stuff between them. So on top, we have sitting our capillary endothelial cell. Here we have our sandwich cell basement membranes. Here we have an epithelial cell that is part of the nephron itself. Right, so we can think of this overall as the glomerular membrane, these kind of 3 layers. But you can see that these gaps are really going to help lots and lots of stuff go through. So those specialized cells in Bowman's capsule, or sorry, in the layer surrounding the capillaries of the glomerulus leading into Bowman's capsule are called podocytes, with extensions called pedophils, basically phi. So that's basically what we're looking at. We have these little foot-like processes, and those slits are kind of between the foot processes, so creating lots and lots of gaps with these slit pores between the pores. Okay, so we'll pick up here next time talking about all about how glomerular filtration works. There will be a little bit of math involved, but it's not too bad, hopefully.